going to share my screen. Okay, so in the previous lecture we dealt with mesoderm and specification of mesoderm into the somite. For this lecture we're going to be looking at the late development of the endoderm. What we're going to be looking at in this lecture? Well, we're going to look at how the lung and the stomach are differentially specified. We're going to look at how along the gut, different specification of the gut allows for different anterior, posterior patterning of endodermal tissues. We're going to compare the liver to the pancreas, and then oddly enough we're going to compare the pancreas to the cerebellum. So, during development, the lungs come directly from the gut, a part of the gut called the anterior gut diverticulum. And this is the foregut of an organism, and you can see progressively over time that this respiratory diverticulum, which is just a name for a budding off organ before it becomes an organ, progressively buds off and then forms the trachea and the lung buds, and you can see they're distinct from the esophagus and pharynx from which they came. How is that tissue identity specified? Well, we've experienced Wnt signalling before, and in this situation we're interested in the inhibition of Wnt signalling compared to the activation of Wnt signalling. In lung development, Wnt signalling drives the development of the lung. This is because the stomach epithelium, the squamous, so the mesenchyme that surrounds the area that's going to become the stomach, expresses high levels of what are called secreted frizzled related proteins. Now we've met these before. Frisbee in our Xenopus development is a Xenopus homologue of these secreted frizzle related proteins, and these block Wnt by binding them in the extracellular space, and this reduces local Wnt signalling. So in our developing stomach area, the mesenchyme secretes these secreted frizzle-related proteins. This stops Wnt signalling, and therefore we have a squamous esophageal epithelia. In the presence of Wnt signalling, these epithelial cells get specified to become ciliated respiratory epithelial cells, and this is due to the presence or absence of SOX2. When Wnt signalling is active, the transcription factor SOX2 specifies respiratory epithelial identity. It's inhibited, and therefore no SOX2, no respiratory epithelia. If there's active Wnt signalling, SOX2 is active, and this specifies the stomach identity as opposed to the lung identity. So let's just recap on our Wnt signalling pathway, a little bit of revision. I know that some of you have written about this in your essays. So we have Wnt binding to the co-receptors LRP56 and frizzled. This activates dishevelled, which inhibits GSK3 beta, which prevents the degradation of beta-catenin, and then beta-catenin can enter the nucleus, and with its co-transcription factors LEF, TCF can activate transcription factor. This is the most famous canonical pathway, and it should also be recognised that there's a non-canonical planar cell polarity, or PCP, pathway that involves Wnt signalling, and this is involved in processes such as convergent extension that happen during gastrulation. So let's look at the anterior-posterior patterning of the gut and how this relates to the expression of different, or the production of different endodermal tissues across the body plan. So here is a schematic of the digestive tract and some of the tissues, the endodermal tissues that get produced along its length. So we have here the thyroid, the thymus, the lungs, the pancreas and the liver. And you can see that across the whole of the digestive tract, different HOX-related genes are expressed. So in the anterior half of the digestive tract, we get expression of SOX2. Remember, SOX2 in the presence of Wnt signalling identifies stomach. Well, SOX2 expression identifies all parts of the anterior digestive tract, as opposed to the caudal-related protein CDX, which you might expect can specify the posterior parts of the digestive tract. The digestive tract overall is specified by the expression of HOXA2, and then we see a selection of different HOX genes specifying different locations across the body plan in a very similar way to how HOX genes are arranged in the development of many organisms, including Drosophila and us. And then we have the central region of the digestive tract, which controls the expression of the biliary processes, the ventral and dorsal pancreas and the liver. And we see specific expression of a number of transcription factors in this region. We see the expression of PDX1 and PDF1A, which specifies the dorsal pancreas and ventral pancreas, SOX17, which specifies the bile glands, the biliary tissues, and then HEX1, which specifies the formation of the liver. So, the different regions of the gut obtain their positional identity through the actions of a range of transcription factors, including a HOX code. PDX and CDX are members of the paraHOX gene family. They evolved from the original clusters of transcription factors that are the evolutionary paralog of the HOX cluster. So these are the original Drosophila cluster, if you like, that turned into the HOX clusters we see in tetrapods. PDX1 regulates pancreas development. The CDX genes, as we said, are orthologs of the Drosophila gene caudal and are expressed earlier in development in the posterior growth zone. So we see our CDXC and CDXA expressed in the posterior parts of the intestine and the digestive tract, and then we see anterior SOX2 expressed in the stomach and esophagus, and finally we get genes like HEX that are expressed in the liver. So again, just reiterating this idea that the HOX code that organizes anterior-posterior patterning overall also organizes anterior-posterior patterning of the endodermal tissues, including the gut and related tissues. So how do we determine stomach versus intestine? And it's not surprising to see that Wnt signalling plays a role. Again, there are differential responses to Wnt that dictate different developmental fates. So if our mesenchymal cells express Wnt, Wnt then, so Wnt signaling defines intestinal epithelium. However, as we recall in our stomach, we get the mesenchymal cells secreting these Wnt inhibitors, these secreted frizzled related proteins, and these bind to and inhibit Wnt signaling, and this specifies stomach epithelium. There's also BMPs, which then define the specific muscle differentiation at the border of these endodermal tissues. So it's a combination of specification of mesenchymal tissues by BMP. BMPs then allow those secreted frizzle-related proteins to be produced through the activity of BARX1. This then inhibits Wnt signaling and defines stomach epithelium. So we can see these important signaling pathways, the bone morphogenetic protein pathway and the Wnt signaling pathway cooperating together to specify the intestinal fate versus the stomach fate. So now let's look at the liver and compare it to the pancreas. Remember that they're in the same region of the developing digestive tract. The endoderm that gives rise to both liver and pancreas is defined by the transcription factors FOXA1 and FOXA2. And it's a combination again of mesodermal tissues, this time cardiogenic mesoderm, so the developing heart in conjunction with the ectodermal signaling, compared also signaling from the notochord, and together these signaling pathways are able to specify whether we have a liver or a pancreas. So near the dorsal endoderm, where the notochord secretes factors, hepatogenesis, the production of the liver, is blocked, and instead development of the pancreas is favoured. The cardiogenic mesoderm and the mesenchyme adjacent to this secretes factors which block the formation of the pancreas, and this gut endoderm expresses alpha fetoprotein and albumin, which are components of the liver. So again, it's the proximity of already existing tissues, such as a notochord and developing heart, that define what's going to happen to the developing digestive tract at that location. So what we have is a cascade of transcription factors that progressively specify the tissues between the three separate fates that happen in this region of the developing endoderm. So first of all, we have HX and SOX17, which serve as our ventral foregut progenitors. So all cells in the developing intestinal tract in that part of the embryo express HEX and SOX17. In the developing heart, we have expression of FGF and bone morphogenetic proteins, and this provides the switch between a liver fate and what's called a pancreatobiliary state, so either forming a liver or the pancreas and bile glands. So where we have high levels of FGF and BMP near to the developing heart, we get reduction in SOX17 expression, and this causes the cells to adopt a liver fate. where we have low levels of FGF and BMP, and instead we have the notochord producing signals. This drives the cells to a pancreatobiliary fate, and through the production of this key transcription factor, PDX1 can then determine downstream fate. And then in the presence of Notch delta signaling, so the cells adjacent to the biliary cells through Notch delta signaling become pancreatic cells. So we get maintained expression of PDX1 in a developing pancreas, and we get maintained expression of SOX17 in our biliary cells, and it's that Notch delta signaling, that juxtacrine signaling between Notch and delta, that sets the fate of those cells that wasn't set in the previous stage where we had pancreatobiliary cells. So PDX1 equals pancreas, SOX17 equals biliary tissue, and HHEX equals liver. And that's all set up by the proximity of the heart secreting FGF and BMP and the notochord producing other related other secreted factors that inhibit the expression of HHEX. So we have this concept of molecular specification where FGF and BMP, that's fibroblast growth factor and bone morphogenetic proteins from the adjacent cardiac mesoderm, drive the expression of liver genes. But the competence, the ability to respond to these genes, is provided by the FOXA transcription factors. These pioneer factors cooperate with the signaling pathways to activate a range of transcription factors that drive subsequent differentiation. Finally, there's a binary fate choice between the hepatocyte and the bile duct governed by local Wnt signaling. So again, we get Wnt signaling and Notch signaling defining specific tissue fate. So we have our ventral endoderm, and through the expression of FOXA1 and FOXA2, this ventral endoderm is made competent to respond to secreted factors and adopt a selection of fates that can either be bile duct or hepatocyte cells, dependent on the status of Wnt and Notch signaling, and therefore the downstream expression of those transcription factors HNF6 and HNF4. So a binary decision is made between the bile duct cells, these cholangiocytes, and the hepatocytes. So again, it's Wnt signaling. Wnt signaling, or the absence thereof, is causing a binary choice between two tissue fates. And if we look at the dorsal pancreas, it's actually the opposite way around. So we have FGF and BMP signals from the cardiac mesoderm in collaboration with those FOXA pioneer factors induce liver formation. In contrast, the FGF2 and activin, which is a TGF-beta superfamily protein, very like the BMP proteins, these are secreted by the notochord. These downregulate sonic hedgehog expression in the endoderm, allowing pancreas formation. So this determines the orientation of the liver and gallbladder versus the pancreas in that part, that middle part of the developing intestinal tract. So in this case, the lack of sonic hedgehog seems to give competence to those gut cells to respond to signals from the blood vessels themselves. And this appears to depend on signaling through CXCL2 in the endoderm, and CXCR4 in the blood vessels. So we need the correct combination of chemokine signals and chemokine receptors to spatially determine competence to form pancreas compared to liver and other related tissues. But it's PDX1 that is the key transcription factor that codes for the pancreas. PDX1 is a homeobox transcription factor that is induced both dorsally and ventrally, giving rise to pancreatic buds. And in this in situ hybridization of PDX1, where again in situ hybridizations we're using an antisense probe to the PDX1 RNA, and that's labeled with an enzyme that can turn a dye from colorless to blue. Wherever we see blue coloration is where we see the expression of PDX1. It's on both sides of the developing gut, and notice its proximity to the veins that provide those CXCR and CXCL signaling molecules that specify the pancreas by causing expression of PDX1. If you knock out PDX1 in mice, they develop without a pancreas. So PDX1 is both necessary and sufficient for the formation of the pancreas. So in summary, we have our ventral endoderm and our dorsal endoderm. In our ventral endoderm, expression of FGF and BMP in high levels in combination with those pioneer factors, FOXA1 and FOXA2, drive the ventral endoderm to adopt a hepatoblast, so a liver type state. The dorsal endoderm is then caused by activin expression and the expression of retinoic acid, which suppresses Wnt and sonic hedgehog signaling, which we'll look at a bit later in the context of somatogenesis. This high level of retinoic acid activates the expression of activin, and then PDX1 in the presence of these pioneer factors, FOXA1 and FOXA2, then drives the dorsal endoderm to become the pancreatic bud as opposed to biliary tissue. So we have these pioneer factors, FOXA1, FOXA2. Then we have the presence of these signaling molecules, Wnt, sonic hedgehog, FGF and BMP, and contradictory retinoic acid signaling, which can then drive cells to a specific binary fate. And just to address this role of retinoic acid in somatogenesis, retinoic acid is present in high levels in the somite and also the anterior presemitic mesoderm. So the area that is already somite and the region of the developing embryo that's about to become a somite. And this opposes that FGF gradient, and so we get that clock of notch signaling occurring at the region where the inhibition of the FGF gradient by retinoic acid is prevented. And we'll look at the role of retinoic acid in development of the somites and development of the limbs in later lectures. So finally, let's look at something that's quite confusing. So we have these concepts of the role of PDX1 in the mouse, and downstream of PDX1, various different pancreatic fates can occur. So the pancreas contains islets with alpha cells and beta cells that form parts of these islets. So from our initial PDX1 expression, the pancreatic bud in the presence of the transcription factor PTF1A become what are called exocrine progenitors. However, with expression of PAX6, TGF beta signaling and NGN3, we get expression of endocrine progenitors. So PTF1A and NGN3 are helix loop helix transcription factors that act in a mutually repressive manner. Upstream of NGN3 is this TGF beta signaling, and downstream of NGN3 we get another binary decision between the expression of PAX4 and PAX6, and this determines the differentiation of these endocrine progenitors into either beta cells or alpha cells. And we're going to explore this, or rather you're going to explore this in your third assessment, which will be open soon on QM+. However, in zebrafish, things are a bit different. So NGN3, or neurogliin 3, in us and in other mammals is required to form a normal beta cell expressing pancreas, and beta cells express insulin. However, you can see in fish that have a mutant NeuroG3, they have a knockout, they still produce cells that produce insulin. And this is because another transcription factor called ASCL1B is expressed at the right place and the right time, and if you like, performs the function of this NeuroG3 protein in mammals. So we can still produce beta cells that express insulin. And this is downstream of Notch delta signaling. So NeuroD1 is active and can be expressed in the wild type fish. Mindbomb is a mutant fish that has no delta Notch signaling. And in these fish that have no delta Notch signaling, we get increased expression of ASCL1B and NeuroD1. There's more differentiation to this beta cell fate. But this knockdown of NeuroD1, and this is shown by NeuroD1 morpholino, does prevent cells that express glucagon. So these morpholinos, we can show with this assay for glucagon a significant reduction in glucagon expressing cell numbers. So knockdown of NeuroD1 in these zebrafish is leading to cell loss. So PTF1A and a different basic helix loop helix transcription factor act in a mutually repressive manner to regulate differentiation downstream of Notch signaling. Remember that Mindbomb effect increased the amount of beta cells. Upstream of the different basic helix loop helix transcription factors is TGF beta signaling, which is what we saw in those previous slides. Downstream of the base of these transcription factors, the homeobox genes PAX4 and PAX6 drive subsequent differentiation of endocrine progenitors, along with the basic helix loop helix transcription factor NeuroD1. So again, we have Notch signaling playing a role in binary specification of cell types. In this case, the binary specification of alpha cells versus those insulin-producing beta cells. So remember, we still had beta cells, we didn't have alpha cells, so we've got differences in our Notch expression, And remember, they can adopt both of these fates until notch expression fixes them in one or other of the fates. This also happens in the cerebellum, oddly enough. And some of the same transcription factors, so the transcription factor ASCL1 is negative in regions where the cerebellum is produced. And this requires TGF-beta signaling in specified domains. So, if we look at a PTF1A expressing mouse, we see very clearly that this key cerebellar factor ATOH1A is repressed when PTF1A is introduced by in utero electroporation. So that's actually going into the developing embryo in the mouse and then zapping the PTF1A, which remember is a pancreas specifying gene, into the area of developing cerebellum. We see less of this ATO1A expression. Likewise, if we introduce by electroporation the ATOH1 gene, we get this reciprocal relationship where PTF1A is repressed. So we have this binary decision making between high levels of ATO1 expression and high levels of PTF1A expression. And this is the same as what we see in the pancreas. We see this same binary decision where we have PTF1A producing the exocrine progenitors, and then in the absence of PTF1A, we get a decision that drives development towards an endocrine progenitor, and then further binary decisions between PAX4 and PAX6 determine the identity of these cells as insulin secreting beta cells or alpha cells that produce glucagon. And in the cerebellum, we see regions with this PAX6 staining. So the same binary cell fate decision that happens in the pancreas is also happening in the cerebellum. This is antibody staining in a hindbrain section of a developing mouse embryo, and we can see this region where the PAX6 staining is present in the developing cerebellum. So NeuroD1 seems to also have a role in the cerebellum. NeuroD1 seems to be important in the pancreas. It's also important in the cerebellum. And this role in the pancreas and cerebellum was determined in this paper, where if you knock out NeuroD1 in mice, it kills the mice too early. However, if you use the insulin promoter to misexpress NeuroD1 in the pancreas, you can get expression only in the pancreas and not in the cerebellum. So then we can use NeuroD1 to determine the role of these binary cell fate, its role in these binary cell fate decisions in both the cerebellum and the pancreas. And in this other paper, where we see this transgenic being used, this knockdown in the presence of insulin, strangely enough, we get far fewer excitatory neurons in the mature cerebellum, and therefore a much smaller cerebellum. So this gene is important in specifying not only pancreatic fate, but also cerebellar fate. So we have our overall model where PTF1A and a different basic helix loop transcription factor act in a mutually repressive manner to regulate differentiation downstream of Notch signaling. Upstream of the different basic helix loop helix transcription factor is our TGF-beta signaling. Downstream of this transcription factor, we see the homeobox genes PAX4 and PAX6 drive differentiation of endocrine progenitors along with the basic helix loop transcription factor NeuroD1. And we see exactly the same process occurring in the pancreas as we do in the cerebellum, suggesting that we have similar developmental signaling pathways that determine the fate of both of these tissues. And so in the cerebellum, we have this binary decision between PTF1A and ATOH1. And when we get expression of PTF1A, we get inhibitory neurons. And when we get expression of ATO1A, we get excitatory neurons. Remember that difference, the binary, we turn on the PTF1A and we get no ATO1 expressing cells. And if we turn on ATO1A expression, we get no PTF1A expressing cells. The ATO1A expression cells then become excitatory neurons in the expression of PAX6 and NeuroD1. But we don't get any of that downstream binary decision making between beta and alpha cells because that's not required in the cerebellum. So PTF1A and ATO1A act in a mutually repressive manner to regulate differentiation downstream of Notch signaling. And upstream of ATOH1 signaling, as before, is TGF-beta signaling. Downstream of ATOH1, the homeobox gene PAX6 drives sufficient subsequent differentiation of endocrine progenitors along with the basic helix loop transcription factor NeuroD1. So the same transcription factor binary decisions are being used in different tissue systems with different outcomes. So that's the end of that lecture. We looked at the importance of Wnt signaling to determine binary decisions in epithelia of the gut, and also how tissues such as the heart and notochord secrete antagonistic molecules, so FGF and BMPs versus retinoic acid, for instance, and these determine the specification of the liver versus the pancreas. And then we looked at how pancreatic cells make binary cell fate decisions with specific expression of transcription factors, and how these binary fate decisions are used multiple times in the body, including tissues such as the cerebellum. So that's the end of that lecture. I'm just going to stop sharing, and I will see you all tomorrow for the early office hour. Do remember to come to those office hours. They are before the lecture. Do consult your timetable, and for the next two lectures. So I will see you then. Okay, bye.